Hi, everyone. I'm Sam Hughes, manager of the Center for Experimental Neurotherapeutics and the St. Jude Children's Research Hospital Translational Neuroscience Initiative. I recently moderated a session at the ASGCT Annual Meeting called To Biomarker or Not To Biomarker, Use Considerations of Cell and Gene Therapy Drug Development. It's a big topic to address, and today we're getting a quick insight from Dr. Rebecca Ahrens-Nicklas. She's a physician with the Metabolic Disease Program and the Division of Human Genetics at the Children's Hospital of Philadelphia. She's also, importantly, one of the 2025 recipients of the ASGCT Outstanding New Investigator Awards. She's here to help us understand when biomarkers may help move cell and gene therapies forward and when they might fall short. For some, biomarkers can just seem too technical, and for others, there may be a misconception if it's the silver bullet to get a gene therapy approval. Our goal here with this community quick take is to make sense of how biomarkers are used, and most importantly, how the voice of the patient advocacy groups is critical to shaping the process. Because really, at the end of the day, biomarkers are just tools to help us answer a simple question: is this treatment doing what we hope it will do for patients? So without further ado, let's dive in. Welcome, Dr. Rebecca Ahrens-Nicklas. Thank you for joining us today.
Hi, thanks for the introduction. It's great to be here today.
Yeah. So really the first question, can you help us understand what exactly is a biomarker and how do they differ from other kind of, let's say, more traditional outcome measures in clinical studies?
Yeah, it's a great question and actually a pretty complicated one. But if we're going to try to narrow it down, you can think of a biomarker essentially as something that you can measure that lets you know how a person is doing. So essentially how their normal biological processes are working, or if they have an illness or a disease, what is the current state of their disease? And this becomes important when you start thinking about trying to develop a drug for a patient, because you want to be able to measure whether that drug is doing something. And so a biomarker may be useful. It's not always useful, but it may be useful in trying to measure how a person is responding to a drug or a therapeutic intervention like a gene or cell therapy. And it's a great question. How is this different than a traditional outcome measure? So if you think about, if let's say you are trying to develop a new drug for a disorder that causes intellectual disability or difficulties learning, right? The goal you're trying to achieve here is to help a child or help a patient be able to meet developmental milestones and learn well, right? So if you think about a traditional outcome measure, it might be measures of how a child does in terms of being able to walk on time or talk on time or learning new things. But those outcome measures are complicated at times. And so what a lot of investigators and what a lot of physicians are trying to do is to figure out if they can measure something quantifiable, either in the blood, in the urine, or some other quantifiable marker that gives you an idea of how a patient is responding to a therapy that's more sometimes immediate and also quantifiable than something complex like measuring a child's development. And so essentially it gives you an indication of how someone is doing and how they might be responding to a drug, or at least that's the goal of a biomarker.
Right. So thinking about that in terms of like how you as a physician, as a researcher, think about it, how it can be used from like the outsider's perspective, like what's the hype? Why should we be excited about these biomarkers, again, whether they're like a blood biomarker or something else like you were talking about, as we think about like cell and gene therapy trials or getting like drugs to patients faster? And like, yeah, so why is this such an exciting thing for people to be thinking about and talking about right now?
Yeah, I think it's an exciting thing because it allows you to sort of have a handle on and try to really quantify whether or not a drug that you're developing may be working, right? So it's something that is measurable, which is so important in the drug development process, right?
Yeah.
We have to be able to show that a therapy that we are trying to develop has the potential for benefit in a patient, right? We put patients at risk by giving them therapy. So we have to be able in some quantifiable way to show a benefit. And in general, biomarkers allow you to measure that benefit more quickly and more robustly than other complicated outcome measures. And this is something that we're all sort of familiar with in our everyday life. As those of us are growing older and have elevated cholesterol, for example, you might take a statin, and then you look at that biomarker of watching your cholesterol cholesterol level hopefully go down in response to that statin. You're not waiting to see if your risk of heart disease or a heart attack is decreased. You have that real-time feedback that because you're taking that cholesterol-lowering medication, your cholesterol number is going down. And so we need these quantifiable measures in order to be able to give us that feedback in real time that something might actually be doing what we hope it's going to do. It's really important that we develop these biomarkers. On the flip side of that, you have to also remember that not every biomarker actually predicts the clinical outcome for a patient. So you have to be very thoughtful about which biomarkers you select and what you're measuring.
Yeah. Well, so that gets to kind of my next question, which is what are like the challenges, especially in rare disease, about like identifying biomarkers, validating them? What does that really mean in rare disease? And how do you really, I guess, from your perspective as a researcher and a clinician, how are you confident that a biomarker is or isn't reflective of a disease process?
Yeah, it's a great question. Now, I think there are some unique challenges in trying to develop biomarkers for rare diseases, but also there's some unique opportunities, right? And I'm a little biased in that I essentially only see patients with rare diseases. But most of the patients that we see with rare diseases, their diseases arise because of a single genetic change, right? So there's something different about their genome that makes them more susceptible or leads to their disease. So we understand for a lot of our rare disease populations a little bit about the science behind their disease. So that allows you to hone in on what type of biomarkers might be useful because you can start thinking through in a scientific way what you would expect to build up or what markers you would expect to be abnormal. So it sort of guides your view or guides where you're looking in terms of how to develop a biomarker. So in the rare disease space, which are mostly rare genetic disorders, that is one huge benefit, right? We know where to look in terms of what type of biomarkers to measure. On the flip side of that, in order to have a biomarker that you can say is definitely abnormal in a certain disease and may or may not respond to a therapy, you have to measure that biomarker in a population of patients. And we all know in the rare and ultra rare disease space, accumulating enough patients and enough samples to really convince yourself that a biomarker is abnormal is really hard. It is the work that so many advocacy group leads, so many physician scientists that I know are trying to do in partnership with advocacy groups. But just to get samples from every rare disease patient with a diagnosis is really hard. And so that's one of the challenges of using biomarkers in rare diseases. That being said, for rare diseases, it's also really hard to do serial neurocognitive testing, for example, or measurement of developmental milestones in a large population. And so having these biomarkers, which, yes, it's difficult to obtain, blood or urine samples from 40 kids from around the world, it might have less variability for many biomarkers than some of these other outcome measures that we've talked about, like neurocognitive testing. And so there are pros and cons, right? There are challenges and unique opportunities in the rare disease space. But I think we are learning as we move forward and a number of cell and gene therapies are either getting approved or submitting their requests for approval to the FDA. Biomarkers have been a huge part of that in the rare disease space. I think that will continue. And again, it's because oftentimes these measures are more reproducible and reliable than other functional outcome measures.
Right, right. So thinking about biomarkers and their use in clinical trials and how, thinking about the biology of the disease as a scientist, you can kind of identify what makes sense, again, kind of biologically. But how do you know, quote unquote, how do you know if that biomarker that makes sense biologically reflects things that the patients and families care about?
I think that's a great question. And I'll say from the outset, you don't know, right? And so that's what a lot of the hard work comes in terms of what we call validating a biomarker, and not just validating a biomarker from a scientific perspective, but I think it's really important to also validate a biomarker in terms of the patient advocacy voice, and whether or not it's a valid biomarker for the concerns of our families and our patients that we care for. And so I would say to validate a biomarker, just to sort of define what I mean by that. What I mean is you measure this, let's say, blood biomarker in a series of patients. You then have to correlate or link the fact that a change in that blood biomarker or differences in that blood biomarker are associated with differences in some sort of outcome that you're hoping to achieve, whether it be, I don't know, the ability to take your first steps or the ability to, do certain activities of daily living, like take care of yourselves as you grow older. Like there's a number of functional outcome measures that are really important. So the validation steps of this is really linking that biomarker to some sort of outcome that is of clinical relevance. Now, where we get the patient voice and the advocacy voice in this is what do we mean by clinical relevance, right? What actually matters to a family? And I'll be honest with you, what I think matters when I meet families in clinic or when I'm sitting in my lab is probably very different than what a family thinks matters at 2 a.m. Taking care of their children, right? And so,
Right.
and we've seen this over and over again as we've thought about biomarker development for rare diseases. So having the patient perspective and the advocacy perspective as a voice and helping guide what we see as sort of that gold standard outcome measure is really important. So having patient-reported outcomes, for example, where families and advocacy groups can get together and really, at a population level, understand in their community what they want to change about the disease process for their child, and then using that to help link the biomarkers that we're talking about. So showing that our biomarker reflects a change in some of these outcomes that are really important to families.
Yeah. I mean, so it sounds like there's a lot of opportunity here for the patient advocacy groups or representatives of those groups or the families themselves to really impact how biomarkers are both like identified and, as you described, kind of validated potentially, right? And so what are things that you would recommend advocacy groups or those people who are like living and breathing this space do now if they're like looking or preparing for their first clinical trial, the first potential drug, or even very, very early in the process? What can they do now to help this process, to help identify biomarkers, to help the researchers understand what is important to them?
Yeah. I think there's a number of different things advocacy groups could do, both on the sample collection and obtaining the sample side, but also in the patient-reported outcome side. So speaking first about getting those crucial, precious samples from patients, we know that getting blood, getting urine, sometimes getting cerebral spinal fluid or CSF, which is a very invasive procedure, is hard for families of kids, especially, I think a lot about pediatrics, but kids with chronic illness, right? And those resources are precious. And so one huge role that the foundations can help with and the advocacy groups can help with is to make sure that those resources are available to researchers in a responsible way, and that no one person is siloing samples. Because those samples are precious, we need to make the most of every sample. And so advocacy groups and families can really help ensure that as they provide their specimens, as they provide their samples, that they are doing so in a way where they can be used by a number of investigators for biomarker development, right? So that's one way that advocates can really make sure that they're using those samples to the best that they possibly can be. On the other side of this, as investigators or as advocacy groups are getting ready to help support clinical trials, let's say of a gene therapy product, they can really start that discussion years beforehand about how to quantify meaningful change in their population. So working with investigators to help derive patient-reported outcomes, which are exactly the things we were talking about, but those quantitative measures of what matters to families. And there's a number of different scales and different approaches and different interview methods that can be used. And I know a lot of rare disease advocacy groups have gone through this process and are willing to help other advocacy groups figure out how to do it. And there's great resources and things like NORD and other resources that really can help guide groups that are trying to do this. But the earlier you do this, the better, because it gives investigators more time to link those biomarkers with these outcome measures. So it's hard if a month before a trial, people decide that we want to measure sleep and that sleep's really important to families, but no one has studied sleep in this population yet, right? And so we don't have a good quantifiable biomarker of sleep. But if we know going in, as we're doing all the drug development studies in the lab and all the safety studies and all the clinical trial readiness studies, which means studying patients with this disease, if we know that sleep is a priority for families, you can build that in at every step of the drug development process. So the earlier that our foundation partners, our patient partners, our family partners can identify which outcomes are really important, the more we can study them in preparation for a clinical trial. So I would say start early and really think about, as a community, what outcomes would be meaningful to you. And you can do that by developing patient-reported outcome measures.
Right. It sounds like it could especially be important for a particular kind of population where things that might matter in the younger children in the population might be different as you get older and as things change and as life changes, and
Correct.
what do the parents and caregivers think
Yes.
and care about. So I think that it seems like there's a broad range of the kinds of information that we can get from kind of the spectrum of patients within a population.
And you might need different biomarkers and different functional outcome measures for different stages of a disease process, right? You might have an infant diagnosed pre-symptomatically with a certain disorder, and the goals of that family and that patient population might be very different than, especially, for example, in a neurodegenerative condition, than a child who has lost neurologic skills, but you want to maintain some semblance of sleep and comfort, right? And you might need different biomarker measures and different outcome measures to capture what's meaningful to those two very different patient populations within this rare disease space.
Yeah.
And so I agree. I think over the course of a disease process, things change, and you need to understand the priorities of the population over the course of the entire disease.
Yeah. Well, I think this has been, super helpful to help kind of distill what are biomarkers, how they're used, how researchers think about them, how patient groups can be involved with the process and as early as possible. Are there any final thoughts, Becca, that you have on this topic?
Yeah, I think it's important to remember there's no one magical biomarker, right? There's no disease really where one biomarker captures everything about the disease and is a perfect measure of how well a therapy works. So I think we have to be realistic about what biomarkers can and cannot do, but realize that they do have the potential to really accelerate the timeline of drug development, and to give us insights into whether or not what we're trying to develop it might be meaningful for a patient.
Yeah. Well, really, thanks again, Becca, for your time and your expertise in helping to kind of educate us and like the ASGCT community and the broader community of advocacy. I know that this is going to be a topic of biomarkers and drug development that's going to be a continuous discussion, a continuous conversation. So really, really appreciate your time, and thanks everyone who's listening in, and hope you're able to learn a little bit more day by day. So thanks again, Becca, and thanks for everyone out there.
Thanks so much. Thanks for having me.